technology. And we've begun. So, you all got that little introductory stuff. So I'm going to go back, and so I have both these PowerPoints loaded, but I'm going to do the Cowan one, and more or less from the beginning, without having to go. I mean, first of all, that's CHONPS. That's important. That's on the test. You got carbon, hydrogen, oxygen. I'll write it down again. You're looking at me quizzically. I know each of you listen to these the night after. That tonight you're going to be listening to this in earnest. You have nothing better to do. Right. Yeah, no, I know. I was saying that facetiously. Okay. So that's on it. And then the other part. So, I mean, I don't care about, they go crazy with this stuff. Essentials, macronutrients, micronutrients. Maybe in your nutrition course, essentials an important definition. We had talked about that. But other than that, really not. And I don't want to go into where we got the carbon from, where we got the oxygen from. It's out there. We got it. They got it. Good for them. God bless. 70%. Okay. Here's the CHONPS. All those types of things. And again, they can do a great deal with very little. I think that's really maybe the most unique feature of all of this, is how rapidly and we see this in life constantly. It doesn't take much to have stuff grow, whether you want it there or not. That's just the nature of the beast. And they give you the percentages, and that's fine. I probably didn't make this clear. Really, the two terms I will ask you about are these two. Autotrophs, very self-growth. So it's all about photosynthesis. So they have to have chlorophyll. So the only one that has chlorophyll in the microbial family is algae. And for our purposes, the only one that has in the non-microbial world, our world, are plants. I'm not going to deal with exotic photosynthetic bacteria or anything like that. I mean, sure, that's where chloroplasts originally came from, that whole endosymbiotic theory stuff, but it's really not that relevant to us. And you saw what it does. Basically, that's where we fix carbon, again, stapling inorganic carbon, typically in carbon dioxide, together to make organic molecules and getting oxygen as a benefit. And so, the energy source comes from the sun. That's what it means. Heterotrophs, different troph growth. Basically, what we do, everything you're working with the lab, every microbe you're working with the lab, they're all heterotrophs. I mean, if you want to get fancy, they're chemo-organo-heterotrophs. They gobble organic chemicals like we do to survive. Very tasty organic chemicals. So voila, here we are. And so I'm not going to go into any of that other stuff. I mean, it's just too much. It's too much information. Eating stones. Okay. Oh, I had a stone for lunch. Oh, tasty, good. Oh, you know what I have in my bag though? I got a scone. I have. No scone fans. See. And they'll talk a little bit about. I don't want to go crazy because that's in another section. Kind of what makes diseases and how they spread, and the difference between microbes that can cause us harm and don't. So the term parasite is something you'll be asking you about down the road, as is the term pathogen. And again, there's a whole lot of them, and whether they live on your body, as many do, or inside of you, which very, very few do. So I don't consider things that are in your colon or in your mouth endoparasites, because they're effectively on your body. You want to think of it that way, because they're either on a dry lining on your skin or on a moist lining as it goes through the alimentary canal or the respiratory tract or the genitourinary tract. I mean, that's an important point, I think, when we start talking about diseases. Intracellular parasites, there are some bacteria, and we explore these on a case-by-case basis. So you'll see all the different ones that are part of it. They're viruses, by definition, are intracellular parasites. Okay? And you'll see that the plague is an intracellular parasite, for instance. And so when we examine those particular diseases in the second half of this course, we'll deal more with that. So I'm not skipping them to skip them. I'm skipping them because a lot of this stuff we go over again. And I never could quite see the relevance of all of this stuff that's here. This is review stuff that you do need to be familiar with. This is where we left it last time, is to understand the significant difference between our cells and effectively all bacterial cells is they have a cell wall. And because of that, it's different in how, and this plays a very important role in how we are able to treat them effectively and remove them if they are causing us harm. Okay? And so this goes over these transport mechanisms. And again, it's all about movement. And again, the driving force for all of these, when you're talking about diffusion, you should all know this: concentration gradient, period, end of sentence. Whether it's water, the solvent, or it's any of the other stuff, concentration gradient drives the bus, so to speak. Higher to lower concentration. Down the road, you see things, learn about in respiratory and AP2, about solubility and being how well easily it moves through a membrane. There's some nuances to that. And hopefully you'll get all of that in AP2. Quick point, if you want a clarification and maybe it's difficult and you want me to help you, an explanation from me, just maybe it'll help you, just shoot me an email. God forbid you drop by the office. It's a mess you can help me clean. So osmosis, selectively permeable. Okay, water basically moves fairly easily till it's balanced. We know what that is. You've seen that. This is the part. So again, without going over it again like we did in AP1, isotonic means it has the same relationship of solutes to solvents as effectively all of our body fluids, be the intracellular or extracellular or plasma or anything like that. The water concentration, therefore, is equal inside and outside. So even though they're at equilibrium, it's always moving, it really stays around the same. Basically around a state. It's a steady state. They say around a set point. It's a good thing. And so equal in both directions, net water movement is equal. So we certainly could not, if you want to think of it, we could not destroy a microorganism by placing it in normal saline. It's just not likely to happen unless it's extraordinarily unusual. If it's hypotonic, and these two are more relevant because of the cell wall. And here are cell walls, and here are those without, like a protozoan, if we're dealing with microbes. Here, with a cell wall, you have water going in. This swells, in this case, the protoplasm, whatever they want to call it. It swells the interior up against the wall. The wall usually prevents the cell from bursting. You can't, water effectively is not a way, large concentrations of pure or distilled water is not effective in destroying bacteria. Hand washing is a different thing. Then the water is playing a role mechanically in moving them. And there may be something like soap or an agent like that we'll get into in the next chapter we do and how it facilitates removing them, okay? Water doesn't, go ahead, water me. They're a plant. Go ahead, water me. Make my day. We did this before. This is different. It will burst. You might remember from A and P, they show you a picture of that, a graphic picture. It bursts so fast in a red blood cell that you can't focus unless you're having a continuous view microscope and watching it. By the time you focus the microscope and you put too much water in a tiny amount of blood, all of those red blood cells will burst. Our cells have very, very little tolerance to that. That's very important. So water is ineffective. On the other hand, hypertonic solutions, overly salted water, seawater, salt in and of itself, that's special. It causes these cells to shrink. And when they shrink, basically the membranes no longer function. They're likely to, they're not going to hold stuff in anymore. They're going to burst in some way, shape, or form. Okay, we got a fancy name called crenation for it. Here, the fancy name, because remember, it's a unit. In a bacteria, the cell wall and the cell membrane are a unit. If you can separate them, in this case, by sucking out the water, you pull the membrane away from the cell wall, it ruptures. Since it's a plasma membrane, we use the term plasmolysis. Okay. And that's, so if we're looking plasmolysis, rupturing that membrane, we're typically talking about a bacteria. If we're talking about an animal cell or a protozoan cell, With it shrinking, we typically use the term crenation, C-R-E-N-A-T. So salt's been around for a long time. Salt's a great preservative. It's been used, it's still used in a lot of things that are there. The bacala fans. Salted. You got to watch more cooking shows. That salted cod from Italy. It's a delicacy. You make these really good little fish balls and fish cakes out of them, but it's packed in salt. The best anchovies are packed in salt. The best capers are packed in salt. No fans of capers, huh? Okay. And so I'm not doing the passive and active transport thing. Go back. Go back. Way back. Active means expending energy, pump, endocytosis, phago, Pinot, did all that. Good to go. See you later. Temperature. Okay. Really, the areas we focus on in this chapter and how they grow, every organism, every microbe, ourselves included, even a full-size organism, a non-microbial organism, have temperature that they optimally grow at. Temperature is different. Most of the time when we talk about things in our environment, when we look at things scientifically, very famously, you see this. You see whatever it is, arbitrarily. Let's say it's pH. Our bodies, you might remember this from lecture, have a pH of roughly 7.35, not 7.47. You might look at this with regard to growth. Growth would be the y-axis, pH. I know it's pH. What happens if this is optimal? We typically this thing. Bell curve, right? You've seen that before. And that is true for pH. Nearly all the organisms that you're going to see, this is, well, get to temperature in a second, with regard to pH, are around, typically grow between pHs of roughly 60. Call them neutral, which is a neutral environment, not acidic or alkaline based on their physiology. Neutrophiles. They follow a bell curve. If it gets more acidic, this would be 0, this would be 14. And if it gets more alkaline or basic, okay, in those directions, it doesn't grow as well. And eventually it won't grow very much at all. And eventually it's going to denature because you have proteins inside. You remember all this. Okay. I use the term with the E, and I emphasize that because in white blood cells, the most common white blood cell in our body is called a neutrophil. That's important. If you want to get confused, I would never have you. Not if putting in a letter, I think that's a little bad. I'm not saying someone else might not do it. I would. And again, you could call me up and say, was this a misprint? This was a typo. So you have that luxury here. So that's pH. It's a bell curve. Not so good with temperature. Temperature, we look at factors like optimal temperature, we call it T optimal, temp optimal, and then we look at temp lethal, when it's been done. And so it starts like a bell curve. If you're looking at growth here, we're just going to show temperature increasing. Okay, T optimal is here. We all know what T optimal is for us, and for basically the microbes that affect us, it's 37 degrees Celsius, good old 98.6. Except it doesn't do this. T lethal is very close to it. Boom! It's like blow off a cliff. That's the unique things about temperature. Why is this so terribly important? The way we typically get microbes in our environment, and certainly in a hospital, is temperature. We superheat them, destroy them, we cook them, get rid of things that could cause us digestive problems or digestive infections, right? Okay. That's all because of this graph. It's not a bell curve. When we freeze them, or chill them. It just slows metabolism. It's not an alternative. You know what happens. I mean, I understand they can spoil in the fridge and even be damaged in the freezer, and we're going to get to that down the road. But effectively, if it's handled properly and you can get to it in time, so to speak, you'll be fine. Not heating. You can't. You heat something, you can't unscramble the egg. You can't make it. I'd like this a little less well done. Good luck, trooper. Buy another steak. You get another burger, right? Doesn't work in reverse. Why? Because temperature, okay, denatures the protein. When you denature, by definition, it is irreversible. Very important point. That's why temperature is so important. It's not a bell curve. And inevitably, in living systems, the optimal temperature is very close to the death temperature. So the death temperature for us, so that's 98.6. If you get, just for instance, arbitrarily, if you get to 40 degrees Celsius, that's 140 degrees Fahrenheit. Or really sick. If you get to 42, you're at 107.6. You're not. You have hyperthermia, it's going to be irreversible, things are going to be shutting down. You're not. On the other hand, and you probably learned about this, I would think, in AP2, there's a lot of evidence, there's a lot of anecdotal cases that demonstrate when somebody, like these days, if they get to somebody who falls in the river or in the pond when it's frozen, you get them out. I mean, you try to resuscitate them, even if they've been down for 30 minutes, 40 minutes out. You try. Because the cold water is limiting the thermal decomposition of the body. Okay? At room temperature, they'd be dead. If they were without oxygen for six minutes or so, they're going to die. They'll be brain dead. Not necessarily so. And if you go into a clinical field, you work in an emergency room, you do that stuff. It's all important stuff. So you have minimal temp, the lowest temp that permits continued growth and metabolism. Stop is different than die. Maximum temperature, highest temperature of growth that can proceed before proteins are denatured. For us, once you get past 104, things get very shaky. Okay? I mean, people are really sick. We'll get to chicken pox. I'll talk about all my 3 kids. They all had the chicken pox. My daughter, and remember, they're not biological siblings. They all had different presentations. My daughter spiked to 106. They were ready. They came in and said, "We're willing to life flight her now if you want down to Children's." We were up in Oil City, Pennsylvania. We're going to life flight her. And we held on and it broke. It is. No snacking. You can snack.
Oh my God, please. I have no break, Dr. Stein, at all.
You what?
I have no break at all.
Oh, I thought you said you're old.
No,
I got old covered for all of us.
I have no break at all. Like,
Oh, I'm giving you a hard time.
this is my snack time. Thank you.
Go ahead. It's all right. I'm a forever snacker.
Oh,
Yeah,
yes.
get me started.
That was easy.
Shh, everybody, hush.
Okay, we'll be quiet, sorry.
So there are 5 categories for you to know. This is where the graph is a little bit better on the other PowerPoint. I cooked that up for you already. See, I was well prepared. There it is. There are 5 categories. This is a little confusing. The ones that are important to us are what we are and what most bacteria that cause us harm called mesophiles. Meso is in the middle. Phile means they like it. Their temperature range is typically between 25 to 45 degrees Celsius. Use those numbers. I'm telling you that for a reason. You probably have guessed that. Okay. Pathogens, typically staph, strep, blah, 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 blah. Basically, they grow at this temperature. Why this is clinically relevant. That's why we're a suitable host for them. Our normal temperature is where they want to breed. For instance, we're an interesting little clinical note. You mentioned we've talked about leprosy a couple of times. Why does leprosy go? We call them acral, acral areas, like the tip of your nose. You see all those the biblical things, or the nose fell off, and the ears and fingers and all that stuff, because temperature is lower. Their temperature is a little lower than 37 degrees Celsius. See, there's science behind all of this. That's the important thing. to understand those interesting differences that are there. We have things that like it warm, thermal temperature, file. We like it. 45 to 70 degrees. Common in hot springs, compost heaps. And if you compost at home, it generates a fair amount of heat. We have one of those tumbler things, you throw stuff in there like that. So there's overlap, as you can see. So there's some mesophiles that are around 40, maybe a skosh above, and there's some thermophiles that are a little lower. But notice the drop off. Notice that falling off a cliff. Demonstrated fairly well there. So where this is relative, sure, it's relative in hot springs. The other place it's relative that's much more common in our environment, hot tubs. So there's certain kinds of problems that we see when someone gets a scrape or something and they get into a hot tub. Okay? That's why you have to be very cautious, and most people do. Just like if you have a pool, whether it's up, above ground or in ground or a hot tub, you should be always checking those chemical levels. You should. Yeah, we uncovered that, but the snow got uncovered because we got a little one sunk into our deck. It's a good thing. Absolutely. I told you, mojitos, midnight, hot tub, good stuff. Hey, I'm human. I know you don't think that. Now, this is where hyperthermophiles, typically archaea, 70 degrees. This is not, you're not going there on vacation. Maybe you're exploring a hydrothermal vent, or you're exploring a somewhat active volcano. Not a great career move, but yeah, that's where you find that. These are different. It's not psycho, psychro. When you put the R in there, it's for cold. This is your fridge, 15 to 30 degrees Celsius. Again, you're going to see a little overlap. Okay, that's there. But make no mistake, there are organisms that can thrive at colder temperatures. Okay, and these are the psychotrophs. Your fridge typically is it's probably around 40 degrees Fahrenheit. You can set them these days depending on your energy usage, but the psychotrophs probably play the major role in spoilage. By chilling it, again, you're really slowing down the metabolic rate, and everything has a lifespan that's in there. I mean, you just can't keep them in there. Some things you can. If you're like me and you like to cook with Asian cuisine and you make your own sauces, oyster sauce, it's like forever because it's got so much salt in it. Just can't. Okay. So you don't like oyster sauce. Did you ever have oyster sauce? See, you try it. You might like it. Okay. And then lastly, in the freezer or in frozen areas, psychrophiles. Psychotrophs mean they grow in the cold. Psychrophiles means they love it. The three that you need to do that really impact us the most, certainly mesophiles, thermophiles, and psychotrophs. These other guys are archaea for all intents and purposes. Okay, got that one out of the way. See, this is fun. The hard one's coming. I've got the two easy ones out of the way already. So here you can see the temperature that they had here. Let me get the other one that we were using. Where did I go? Here we are. So here's Cowan, and they give you a description. Okay, freezer, interestingly enough, freezer burn is less likely microorganisms than mishandling of the way you freeze things. You've got to know how to freeze things properly if you want them to last a long period of time. We'll talk about that in the next chapter, but people just throw it into a zip top bag and toss it in the freezer. Not necessarily the best way to freeze things. The toss, particularly. We'll get there. So it has a little discussion on psychrophiles, rarely pathogenic, psychotrophs. As you can see, Staph aureus, and this is a real bad boy. All you have to do is every time you see people are sickened with food poisoning and somebody starts dying, and it's pregnant women or people with immunosuppressed or elderly, watch out for this. Listeria monocytogenes is a gram-negative rod that's frequently an animal fecal contamination. It's from handling. Okay. There was once one if you like cantaloupe, most people don't think about this. Oh, it looks nice from the store. They don't wash the outside. It was on the rind. When you take a knife and you haven't washed that cantaloupe on the outside, you cut it in, anything that's on the outside goes on the inside. because the inside's sterile. So I'm not telling you to give there, instead of take it into the shower, but give it a good rinse off and some dirt, just get something, get one of those neat little, pads that we all have with the rough end and the smooth end and all that stuff, whatever they are, and do that. A lot of people got listeria and some people died from that. That was all. They just happened to have contamination on the exterior. How it got there, was the soil contaminated by animals, wasn't clear. Be watchful of this. And we'll talk. I mean, it's my little thing about the lettuce. See, you're college students. Now, I know about you in dorms. I know they won't let you cook, but how easy is it to go to the store and get a bag of lettuce and get some dressing and get some veggies and stuff like that and throw it on there, right? It says pre-washed or triple washed. Did you see them wash it? I didn't. Get a little colander and wash it. Better yet, get a salad spinner, a lettuce spinner. Love it. That way the dressing holds on nicely. You don't want wet lettuce, you want dry lettuce. Holds the dressing, it embraces it. No salad spinner fans? Shh. Oh, we don't leave home without it. We go on vacation, we take the Brita pitcher, we got the salad spinner, we're ready to go. Hey, we take it away.
She moved in my house and made us buy a Brita.
I don't blame her.
She wouldn't use our tap water.
Oh,
She refused. Love my Brita.
We're simpatico. I lived at an apartment in, this is the ends of the earth, McCandless Township, about walking distance to North Hills Pacific Hospital. You couldn't drink the water. It was perfectly safe. It was absolutely. Moving on. So here's mesophiles, 20 to 40, everywhere. Human pathogens, optimal, 30 to 40. They call them thermoduric. Some are more, and again, endospores are heat resistant. Thermophiles, optimally temperatures greater. You can see where they don't impact us as much. Because once you get to a certain temperature, it's pretty much over. And so this is their chart. I didn't love it for that reason. This is the other important one. pH was in there and we covered it. What I think is really important. What is really important is this, okay? It's oxygen and carbon dioxide. Oxygen, we classify it based on oxygen. Those that use oxygen, and remember we talked about this when I talked about those peroxisomes, they can detoxify it. Those that neither use it nor detoxify it, and those that do not use oxygen but can detoxify. They make a lot of noise about this. There are 5 categories, okay? And this is the whole things. The big problem, there's a couple of them. The superoxide, that O2 with the minus sign, is nasty. Hydrogen peroxide, potentially nasty. There are a number of these toxic forms of oxygen. That's why living things that have oxygen in their environment or need them to grow have to have means, like I put on like our peroxisomes. Remember, superoxide dismutase catalase. That's where this is coming from. Here's the reaction. I drew it. They put it on there. It doesn't matter to me. So what happens, you get O2 with a minus, and they probably get this from water in the presence, and it's an enzyme. Enzymes we use over and over again. We call it SOD. It makes hydrogen peroxide still a problem, and oxygen. And then the famous one, hydrogen peroxide in the presence of the other enzyme catalase, light bubbles, oxygen gas. Be familiar. I would think for lab you would need to be familiar with this. That's me. So how do we classify them? We have 5 different categories. We have aerobes. They utilize air. Use gaseous oxygen in their metabolism, possess enzymes they need, like superoxide, dismutase, and catalase. So an obligate aerobe, us, has both of those. So most fungi, protozoa, and a lot of bacteria are obligate aerobes. I mean, tuberculosis grows in your lungs. No better place to find oxygen. These are interesting. They're called microaerophile. They have enough to fight oxygen, but not in large amounts. Micro means small. So normal atmosphere, and remember our atmosphere is 20% oxygen. It has to be somewhere between 5 and 10%. So it's somewhere between, you know, let's say arbitrarily 20 to 50% oxygen. I mean, you and I could survive, but with some difficulty in that, but we could survive. These organisms that live in soil, water, and mammalian hosts, not directly exposed to atmosphere. Here's really big players. Helicobacter pylori. That's the one that's been associated with ulcers. In particular, that's been associated with gastric cancer. Borrelia burgdorferi is Lyme disease. Okay, they live inside. Borrelia burgdorferi live in the bloodstream of insects, but they don't do well with excessive amounts of oxygen. They have limited amounts of those agents to deal with them, and so there's, I mean, they're significant, and there is. When you hear periodically about this disease we'll talk about called necrotizing, and we talk about wound fasciitis. This is the notorious flesh-eating disease. And there's maybe probably less than 25 cases in the country annually, but they make big news. You know, somebody got a fish hook stuck in their finger, and all of a sudden their arm had to come off completely. Neck fash is very, and it actually is caused by a variation, a cousin of the strep throat strep, Streptococcus pyogenes, that happens to be microaerophylla. And that's where it gets it. You'll see that typically we see that in people who have insufficient oxygen going to an extremity. I have seen 2 cases. I'll go over them again when we talk about them. One in a diabetic in a leg. They didn't have a lot of circulation. One in an injectable drug user. Most people rarely see one. I have a really good anecdote about that. Interestingly, that was, if you watch The Pit, they just had a case like that, which I called from when the patient walked in. I always go, how'd you know that? I saw it. Moving on. Facultative anaerobes. They live without oxygen. Okay? So they don't require oxygen, but they use it. I have the wrong one. Facultative anaerobes do not require oxygen, but use it when it is present. E. coli, Staphylococcus. Facultative, when something is facultative, it means it has the faculty, not teaching faculty, faculty or ability to thrive. Okay, so they can what they can do in oxygen, they use it like we do, but if the oxygen is not there, they ferment. And those are those funky smells you're going to get out of the incubator. Have you started to get some of those in the lab? Yeah. That's called fermentation. Yeah, not if you're making sourdough bread, really good. Okay, but if you're dealing with bacteria, yuck. So that is a lot of those facultative, and you're going to see and these we use them in the lab for study all the time because it's so easy to grow them. If you have oxygen, they grow fine. If we're in an area where you don't have as much oxygen, they grow fine. Okay, anaerobes. Okay. These cannot deal with it. They have neither superoxide dismutase or catalase. We call them obligate anaerobes, not facultative. You know, facultative anaerobes can go in either direction. Obligate, these are things we call strict anaerobes. They're obligated to be anaerobic. They lack the enzymes for processing toxic oxygen and diet and its presence. That's why we utilize things. Oral bacteria, for instance, intestinal, C. diff. Only flourishes, that's an anaerobic environment, if you did not know that. We talked about tetanus and botulism. They only grow in an completely anaerobic environment. They are destroyed by air. So there are a lot of these obligate and that's why we utilize oxygen therapy in a lot of things. We put people in what are called hyperbaric chambers. It's like a diver who gets the bends. They're using compressed air. But in a hospital environment, we can use hyperbaric. You can use the same kind of a chamber and do that. So particularly in areas where they have a lot of marine biology and things like that, universities will have that, hospitals will have that. So things that are utilized a great deal in that regard. So those are all things you might end up becoming familiar with depending on where you work and what you do. So and again, they don't have the superoxide dismutase, they don't have the catalase. Aerotolerant anaerobes. Okay, do not utilize oxygen, but they can survive. This is when I said they don't use it. They have a limited amount, okay? They have it, so mainly because they possess alternate mechanisms for breaking down peroxides and superoxide. Most of the time, they don't have the superoxide dismutase. They probably have just catalase in them. And there's ones, and you'll see this. This is why, no, they don't have the catalase. I beg your pardon. I think all they have is a superoxide dismutase. Lactobacilli we'll talk about because they play a pivotal role in female reproductive, and streptococci, and even some Clostridium. Clostridia to me are more likely strict anaerobes, but not when they say Clostridial species, it's not all of them. And you'll see, and we used to do this experiment. We don't do it in this school. Again, you're going to see organisms that grow only at the top. Bacillus, when you grow B. subtilis, it'll grow at the top of the tube. We don't have microaerophiles. You have to use a special growth media for that. But you can see they grow below because they don't want to grow exactly there. E. coli, that tube will be filled. When you start working with the tubes, what you do, the stuff settles. It's like anything else. You take it for a spin. You spin it or you agitate the tube a little bit and you'll see it fills up. Anaerobes will grow at the bottom. Aerotolerant will grow all the way through. And they're ones that, like capnophilic, means they do better at carbon dioxide. These are hard to grow or have to be in special environment. Again, you have a lot of carbon dioxide in your lungs. Streptococcus pneumonia. Most of us, about a third to a half of us, have this normally in our throat. We can grow it out of our throat. Brucella is a little unusual. This, we have Neisseria species again in our throat. Our throat's like a cesspool. We'll talk about that when we talk about throats and strep throat and things like that. Neisseria, these are two major players. Gonorrhea and meningitis. Very, very hard to cultivate in the lab. So it's not something you typically work with in the lab. You need special augers, special growth media to be able to do that. It's very, all the augers are very interesting. So a lot of times we have to make the diagnosis with smears and stain. And they're unusual. Here's the pH one that I was talking about. I don't want to go crazy and go over and give you all the names and all. These are oddballs. Proteus you're going to work with. In other words, normally it likes it alkaline, but it can neutralize the alkaline conditions to neutralize urine, okay, and colonize. Urine's usually a little acidic, and they do cause urinary tract infections. And they don't mention the one I was talking about, the neutrophiles. These are some oddballs. So things that grow in seawater, osmophiles, and it's not the only area. The most famous ones are halophiles, and really, this is the big player. Staphylococcus aureus, you're going to have to know. So you had lab yesterday. What did you guys do? Did you gram stain yesterday?
Yes.
Okay, so you aren't there yet. When you get to what are called selective and differential plates, you're going to grow Staphylococcus on what's called MSA. It stands for mannitol salt agar. That's the way they test it. If you're in the hospital, if you're working in the hospital, or you have a patient and they want to see if it's MRSA, they have to grow it in an agar, a media, that's about 7% salt solution. That's here. So MSA is what's called a selective and differential media, which means it not only does it grow it because of the salt, but it changes color in the presence of certain species, like Staphylococcus aureus. It goes from red to yellow. It's very attractive. You'll like it. So there's lots of different ones in this category, but they are a halophile. So why do they grow on our skin? Our skin is salty. Well, that perspiration, when it evaporates, it's the salts. Radiation, we will talk about more about radiation, much more so when in microbial control, why we use them to rid bacteria or to sterilize. Okay. Plays a very important role. And I don't really get into the ones that are in barophiles, meaning it has to do with pressures and very adaptive to high pressures. Bottom of the ocean would be an example of that. Again, they're oddballs. And so there's a lot of different things, shared habitats, odd things that I don't get into. These are some of those definitions. And what we'll start with, I'm going to go over these. This is the last, and perhaps I'm going to jump ahead and then come back. And not so much biofilms or things like that. It's this. It's this is called the bacterial growth curve. I have to spend a significant time. I would not consider starting it today. This is really super important. So I'm going to dedicate most of Friday to going over that. And obviously be recorded because I know people travel for sports and things like that. But that's the day it's coming. And when we finish that, that will conclude the material for the first exam, which is kind of going along plans. But this is what we talked about. Every book puts them in a different thing. We have a symbiotic is a relationship, a partnership of living things. It's not always friendly. I don't distinguish between mutualism, mutually beneficial, or commensalism, which basically means one thrives and the other one isn't harmed. And that's what they talk about bacteriologists. The bacteria are thriving and it doesn't hurt us. Mutualism is like the remora and the shark are kind of getting away. You have all these examples biologically that you make. I don't really want to distinguish. The big deal with both of these is the host is not harmed. What makes a true parasite is the host organism provides the parasite or parasitic microbe with nutrients and a place to live, and the host suffers. That's a bad guy. Oh, here, live on my skin. You're hurting me. That's a true. So we use the term parasitic relationship for when the microbe or the organism thrives and the host, us, is harmed. You're going to see down the road when we talk about that's parasite. Think of that as parasite with a big P. When you're going to see parasite with a small p, when we start talking about infections like malaria, that's a eukaryotic organism. Okay, Trichomonas vaginalis. A lot of times we call those in our language, oh, they have a parasitic infection. They have a stomach parasite, like a worm. So that there's the definition of what a parasite truly is and sort of the vernacular we use in medicine as to whether it's a bacterial infection, a viral infection, or a parasitic infection. And that's another good thing I think for you to be familiar with. And again, there's a lot of antagonism that goes on in these communities, and that's what's going to happen. When you look at this growth curve, we're going to talk about how antibodies form and antibiotics form. Okay? What they do is they say production of inhibitory compounds such as antibiotics, when the environment becomes unsavory, gives them difficulty in growing, if it's overly competitive, if it's harsh, that's when those that can form antibiotics do. And so whoever gains a competitive advantage by doing this is more likely to survive. And that's why we look so closely, particularly at soil microbes, because there's so many microbes in the soil. We're constantly going to environments, sampling these and seeing if they make new antibiotics for us because we're running out. And I'll leave you with that. And synergism is that the parties will together they're stronger. So I'll go over synergism more thoroughly when we get there. Okay. Thank you very much.